Hello there. This video will cover the topic enzymes for Module 1 CCEA Biology. Um, I'm going to try and go through the content of this topic fairly quickly, picking up on the most relevant areas with respect to questions and looking at appropriate questions for each area. To get most benefit from this video, you need access to a set of my notes and a set of questions, a set of past paper questions. Both of which, if you want those, then you can get in touch with me by text there on my mobile number. Okay, I'm going to move fairly quickly. It's a relatively long topic too. So I'm going to move quickly. Okay, so enzymes then. At GCSE, you would have been asked what an enzyme was, and the answer was it was a protein and a biological catalyst that speeds up chemical reactions. Notable point there was that it was a protein. From the molecules topic, you probably already appreciate that enzymes are large proteins, and they're large globular proteins. Globular refers to the fact that the enzyme has a rounded or three-dimensional shape like a globe. It has a tertiary structure, and the shape of the enzyme is very important with regards to the function of the enzyme. The shape of the enzyme determines the shape of the active site, and the active site then is complementary to a specific substrate molecule. So enzymes are proteins. Enzymes are large proteins. That also brings into consideration how enzymes can be denatured or damaged. Large proteins can be damaged because if you can break the bonds that are maintaining the tertiary structure, that is hydrogen bonds, ionic bonds, covalent bonds, disulfide bonds, and hydrophobic interactions, you can break any of those bonds, then the protein will lose its three-dimensional shape, and that's very relevant then for enzymes, which if they lose their three-dimensional shape, they lose the shape of their active site, and therefore they're no longer functional as they can't fit their substrate. Just notable from the front page there, just to draw to your attention that enzymes are involved both in build-up and breakdown reactions. Tend to associate enzymes with breakdown reactions, hydrolysis reactions, which you've learned about in the molecules topic. Enzymes at GCSE were molecules that broke down large food molecules to small food molecules. That was proteins, for example, amino acids, and so on and so forth. So you associate enzymes with breaking down. Enzymes are also involved in build-up reactions where you're joining molecules together or creating bigger molecules. For example, if you took two amino acids and join them together to get a dipeptide, there's enzymes involved there. So enzymes are also involved in build-up reactions as well. Okay, I'm going to look then on page 2 here. I'm looking here at how enzyme action. I'm going to refer you specifically to question 2 in the question booklet there, which is about an enzyme called ACE, which is an unusual name. And just to note there, in exam questions, you will often come across an unfamiliar enzyme, one that you haven't heard of before, because you don't really know the names of many enzymes. At GCSE, you'd have learned that there was protease is breaking down proteins, amylase is breaking down starch, and lipase is breaking down lipids. There are lots of enzymes involved in all aspects of cells and biology, and here you're introduced to an enzyme called ACE. And a wee bit of crossover with this, because it deals with a module 2 topic. Is talking about high blood pressure, something you might have mentioned maybe in module 2. It's talking about hypertension, and it says the molecule, the reaction is angiotensin 1 being converted to angiotensin 2, and ACE is the enzyme. and it asks you with reference to the mechanism of enzyme action, describe how ACE, the enzyme, converts angiotensin 1 to angiotensin 2. Now, this is bringing to our attention, and this is a common question, where they're asking something about basically how do enzymes work. And you remember back to GCSE, you would have simply said the enzyme and the substrate combined, you get an enzyme substrate complex. The enzymes would collide with the substrate. Maybe I'd have said the enzymes would collide with the substrate. And the higher the temperature, the faster the enzyme and the substrate moved, and therefore you get more enzyme substrate collisions, and you get more enzyme substrate complexes formed. Now, at AS level, however, you learn that it's a wee bit more complicated. that theory put forward at GCSE was often referred to as the lock and key hypothesis, and essentially it holds scientists' understanding of enzymes is based essentially around the lock and key hypothesis, the idea that the substrate is a specific complementary shape to the active site and it fits into the active site, and only then, when it does fit into the active site, does the reaction take place. The so-called lock and key hypothesis. There is, however, a more modern hypothesis, a more modern understanding by scientists of enzyme action. They're still trying to figure out enzyme action. Bear in mind, you can't see enzymes and you can't see what they're doing. So this is really only a kind of a theory or hypothesis. More modern theory is referred to as the induced fit theory. Induced fit theory. And the induced fit theory is essentially saying the same as the lock and key hypothesis, that the substrate does fit into the active site of the enzyme. But here there's an emphasis on understanding that the fit of the substrate into the active site isn't a perfect fit. That actually, for the substrate to get into the active site, it has to, as it were, squeeze into the active site. So it's like putting your foot into a very tight shoe, if you can imagine that, a shoe that's one size too small for you. The substrate has to squeeze into the active site. And when it does so, you would then say that it's a tight fit, and you would then say that the enzyme puts pressure on the substrate because it's squeezing the substrate because the substrate's a tight fit. And that then would cause bonds to break or to be made, depending on the type of reaction. You'd have to mention that bonds are either broken or made in the substrate or substrates to create the product. And then lastly, you would need to say something like that the product, once the product is formed, the product is no longer the correct fit for the active site. The product doesn't fit into the active site like the substrate does. And so the product then emerges from the active site, or just pops out of the active site. I like to imagine it popping out because it's not the right fit in the product. And so essentially that's the basis of the induced fit theory. Now, looking at that question 2 there, that question 2 asks for 3 marks to describe enzyme action with regards to this enzyme is. Now, the first mark would be something along the lines to say that the substrate angiotensin 1 is complementary in shape to the active site of the enzyme and fits into the active site of the enzyme. Now, I would also mention, I would say it's a tight fit. It squeezes into the active site of the enzyme, and when it does, then the next thing to say is that the active site is actually the active site has to change shape to allow the substrate to get in there. So you'd say the active site changes shape as it molds around the substrate. This idea of the active site molding around the substrate, having to change shape and mold around the substrate. And then you would argue that if the substrate's squeezing into the active site's squeezing back on the substrate. You'd say then that the change in shape of the active site squeezes the substrate, and that squeezing of the substrate causes bonds to be broken or to be made. You don't have to know whether the bonds are broken or made, but you have to mention that bonds are either broken or made in this circumstance. And then to get the last molecule, you have to say that the product produced has a different shape to the substrate, and the product it doesn't fit the active site, and so it pops out of the active site. It's released from the active site. So there you are. There's the 3 marks for that kind of question. So if you get a modern day question on enzymes, will expect you to talk about the induced fit theory. You may, however, look at older questions that have been asked in past papers in previous years, and you may find that the old lock and key hypothesis essentially answers those questions. And so this is a developing theory in the last 20 or 30 years. The understanding of science of enzyme action is developing. So modern day questions talk about the induced fit theory and talk about all those elements I have mentioned there. I'm just looking at page 6 in the structure of an enzyme, and I just emphasize a picture there of an enzyme. That picture of the enzyme on page 6 is just to emphasize that an enzyme is a large protein, and often, typically maybe, an enzyme is made of just one polypeptide. Therefore, you'd say the enzyme's not a conjugate. It doesn't have quaternary structure. It just has the polypeptide just has primary, secondary, and tertiary structure, and you can see in that polypeptide there that you can see the secondary structure. I think you can just about see the coiling in the polypeptide. That would be the alpha helix structure, and then the fact that the polypeptide rolled up into a ball shape or a globular shape, that's its tertiary structure. And it's got an active site, then can be some position on the surface of the enzyme. Okay, so denaturation of enzymes then can happen due to high temperatures or extremes of pH. So the enzyme works best at an optimum temperature, and above that temperature it's denatured. Equally, the enzymes work best at a particular pH, and if the pH moves away from that optimum pH, then the enzyme is said to be denatured as well. Its structure changes. So we want to talk a wee bit about those sorts of things. So the next section is just looking then at enzyme activity. Enzyme activity is just essentially a measure of the rate at which the enzyme is carrying out its function in the reaction. And there are several factors then that affect that, as I already mentioned: temperature, pH, substrate concentration, and enzyme concentration. Now, the most appropriate question to deal with this, well, maybe not the most appropriate question, certainly an easy question, is an essay question, or question 9, which asks you to identify from the graphs. You get 3 graphs. First graph there is for temperature. So you get 3 graphs, and you're asked to identify the independent variable. In other words, that's the factor that's causing the change in enzyme activity. The independent variable, of course, the variable that is changing. And you see there the 3 graphs. Just looking at question 9, they're asking you to identify the independent variable, describe the trend, and explain the trends described. So for temperature, essentially we're saying, you can see the graph rises up to a peak from a low temperature, and you can say as you move along that line towards the peak being the optimum temperature, then you can say the enzyme and the substrate have more energy, more kinetic energy due to the higher temperatures. That means they move around more. That means they collide more often, and more often they meet up or collide. And the more frequently they collide, the more, as it were, enzyme substrate complexes form, and therefore the faster the reaction. Beyond that optimum temperature, which is the peak there, then you're arguing that the high temperatures damaging the enzyme. The word is denaturing the enzyme. It's breaking bonds. So you can mention all those bonds. It breaks hydrogen and ionic and disulfide bonds in the enzyme, and that then allows the enzyme, cause the enzyme shape to change, and the shape of the active site changes, so it's no longer so complementary in shape to the substrate, and the reaction rate slows down. Looking at pH there, you can see that the pH graph is essentially, and that's in question 9, that's the second graph there, pH graph, and you can see that there's very definitely an optimum pH at which the enzyme works best. Enzymes are sensitive to pH. The structure of the enzyme then can be changed or damaged by what you call extremes of pH. Extremes being pH is away from the optimum. So if the optimum is 7, then extremes would be 8, 9, 10, 11, 12 in one direction, and 5, 4, 3, 2, 1. Acidic pH is in our direction, and both those directions in the enzyme is damaged. Now, we can say that it's bonds that are damaged by these extremes of pH. It would appear, however, it's mostly it's ionic bonds that are damaged here. But in exam questions, you can simply say bonds are broken by extremes of pH, and that causes the three-dimensional shape of the enzyme to change, and therefore the shape of the active site to change. But strictly speaking, it's ionic bonds. The effect then of substrate concentration on enzyme activity, you can see at the bottom of page 9. At the bottom of page 9, so basically you have enzyme, substrate concentration and activity. Essentially you get a graph. We're getting a graph that's going up essentially that and leveling off. The graph levels up. Describing that graph, you're describing the trend there. You're saying at low substrate concentrations, the enzyme activity is low. And as substrate concentration, you get identified like two areas. That's area A and that's area B. In area A, as the substrate concentration increases, the enzyme activity increases. And the argument here is you could say that in this region, and you don't have to say this, you could say substrate is a limiting factor. In other words, you're saying that there's plenty of enzymes, but there's not enough substrates to fill all the active sites quickly enough. So if you increase the amount of substrate, then you can see the available active sites have substrate then available to them, and the reaction goes faster. Once you reach this point, then you're saying that increasing the substrate concentration beyond that point. Beyond that point, there doesn't seem to have any further increase in enzyme activity. And the argument here is that at that point, all the enzyme's active sites are occupied, fully occupied, and adding more substrate has no effect on the production of product essentially, because all the enzyme active sites are occupied. However, how could you get the activity to go faster? And the answer is if you added more enzymes. So C here would represent if you added in more enzymes, then you've more active sites available, and the rate of reaction, the activity could go a bit higher. So often you'll see a graph like this. Using the idea of limiting factors, we said substrate was the limiting factor down here. If we go to B here, in graph B, we would say that in that region, enzyme concentration or enzyme active sites is the limiting factor along there. And then go up to C once again, enzyme active sites is a limiting factor at C with reference to maybe getting the graph to go a bit higher. So that's enzyme substrate concentration. Essentially the same graph if you have enzyme concentration along the bottom here. If you put enzyme concentration along the bottom, and it's essentially the same graph. I'm just going to put enzyme concentration. Essentially the same graph. So you're basically saying down in this region, in region A here, you're saying the reaction is increasing because as you increase the amount of enzyme, then there are more and more active sites available to combine with the substrate molecules. Just remove this other. If you come up into region B here, then you're saying at this point, increasing the enzyme concentration doesn't seem to have any further increase in activity, and this is because at this point, then substrate becomes a limiting factor. You've got plenty of enzyme active sites at this point, but you need to increase the amount of substrate that's present to get the reaction to go any faster. So essentially, if you increase the amount of substrate, then you could get the reaction going up to higher levels. So enzyme, substrate, and enzyme concentration. Just looking for questions relating to that. As I said, question, I think essay question 9 and 10 both deal with that, 9 and 10. Question 10 has a wee bit at the start of it, the first section, which is only, it's only worth, the first part is only worth 4 marks, where it's asking you about asking about this last graph here. You have to explain the graph. Just question 9 and 10. Now, moving on then to look at cofactors. And this is page 11 in the notes there. Cofactors. So, cofactors. Cofactors. Ask a definition of a cofactor. What is a cofactor? And you'll see there, I think there's a particular question. I think it's question 4. So if you look at question 4 there, you'll see that something you need to know there is the definition of a cofactor. So in question 4, definition of a cofactor. I think let's have a quick look at question 4. So it's a cofactor is a molecule, a non-protein molecule. So it's not a protein. It's a non-protein molecule that needs to be present for an enzyme to work efficiently, for an enzyme to function. Cofactors, so we think of them in a positive light. They are molecules that need to be present for the reaction to actually happen at all. So if you've got the enzyme and the substrate present and you don't have the cofactor, the reaction won't happen. Cofactor. Given some examples there, I've said zinc ions and copper ions. They tend to be things that you've come across that you talk with respect to a person's diet. You would say that we need a certain amount of minerals in our diet to make our body function properly. There's no particular topic in biology where you look at the use of these minerals, but minerals like, and you see them there, like zinc ions, copper ions, magnesium ions, manganese ions, selenium, there's a whole lot of them that are needed in very small amounts to make enzymes that are in your body work well. One that always comes to mind is there's an enzyme in your saliva called salivary amylase that breaks down starch. That enzyme only works well if there are chloride ions present in your saliva, in your mouth. Chloride ions. Now, how do cofactors work? Well, this is my sort of understanding of cofactors. Imagine that's an active site of the enzyme. That's the enzyme. That's the sort of like a GCSE version of what an enzyme looks like with its active site. And imagine this is the substrate. So there's the substrate. Now, you can imagine that how a cofactor works. At the moment, you can see that the substrate fits into the active site, but it's not a good fit into the active site. So you need to introduce the cofactor as well. Now here, imagine the cofactor then has a shape that maybe cofactor sort of looks like that, sort of wedge shape. Well, here's an understanding of how cofactors work. Is it possibly the cofactor attaches into the active site of the enzyme? Like we understand it attaches into the active site of the enzyme in such a way as to make the active site a better fit for the substrate. So that's one way of thinking about it. And the cofactor may be a molecule that attaches to the active site of the enzyme, making the active site a better fit for the substrate molecule. Or alternatively, if you think of it this way, maybe the cofactor could attach to the substrate. Cofactor attaches to the substrate molecule, making it, the substrate molecule, a better fit for the active site. One way or the other, it's about getting a better fit between enzyme active site and substrate. Okay, questions relating to this question 4 then. To define the term cofactor, cofactors non-protein substances that some enzymes need in order to function and be okay. Question 4 then asks you about the structure of the protein collagen. Just for completion sake, collagen, you come across collagen. Collagen is a fibrous protein made up of 3 polypeptides wrapped around each other to create a long protein, a stringy protein, and there are cross linkages between the individual polypeptides called hydrogen bonds, and that gives the collagen its high tensile strength. So that's related to its function. Its function is to have high tensile strength as is needed in tendons and ligaments. Okay. The rest of this question then give you a table, and it's asking about, it's rather a strange question. It's talking about enzymes involved in an enzyme called collagenase. Collagenase. Notice that enzymes, as you remember, the GCSE enzymes tend to be named according to their substrate by simply taking the substrate and adding ASE to the end. So in GCSE you had protein. If you took away the bit at the end, you got protease, lipase. So this idea of adding ASE. So this collagenase breaks down collagen, and you can see what's happening there. They've done an experiment where they have the coenzyme zinc, in this case, present or absent, and describe and explain the trend shown in the results there. So you can see, first of all, describe the trend that's present there. So you're basically seeing that the average collagen fibril diameter is decreasing when zinc is present. When zinc is present, decreases quickly when zinc is present, and doesn't appear to decrease very much at all when zinc is absent. So that's the describe mark. So essentially, when the zinc is present, the average collagen fibril diameter decreases over a period of 25 minutes, over a period of time. The explanation then, you want to then say that the zinc essentially is a cofactor. So you could say things like the zinc makes the active site of the enzyme more complementary in shape to the substrate collagen. So if the zinc fits, so that fits, the collagen fits better into the active site, and so if collagen fits better into the active site, you'd say that collagen can fit better into the active site. And you could say it this way, you could say there are more enzyme substrate complexes formed, or that the collagen gets broken down faster. Part C asks, it asks then about how a therapeutic drug could treat arthritis. Okay, coming on to that then, so this is looking at something else. This is looking at the second aspect of enzyme action, and that is the fact that enzymes can be slowed down or inhibited. So just in the same way as enzyme action needed cofactors to make it happen or to speed it up, equally you can have other molecules present around the enzyme that slows down the enzyme action, and they're referred to as inhibitors, enzyme inhibitors. Enzyme inhibitors then are basically chemicals which slow down enzyme action, and they fall into two groups. Enzyme inhibitors are either competitive inhibitors or non-competitive inhibitors. Competitive inhibitors essentially are molecules which are similar in shape to the normal substrate, and they're so similar in shape that if they're present around the enzyme, they will fit into the active site equally as well as the substrate. So essentially they're competing with the substrate for the active site, and as a consequence, if they're competing with the substrate for the active site, it means the substrate doesn't get into the active site as often as it should do when the inhibitor is present. The other notable thing is to notice that the amount of inhibition, or the extent to which the inhibitor slows down the enzyme action, will depend on the relative amounts of inhibitor molecules and substrate molecules. What that means essentially is if you've only got a few inhibitor molecules present, a low concentration of the inhibitor, a few inhibitor molecules present, and a lot of substrate, then that won't have a noticeable effect in enzyme action, because the substrate molecules, if they're there in the majority, they're more likely to get into the active site. And vice versa, then if you've got a lot of inhibitor molecules relative to the amount of substrate molecules, then the inhibitor will have a noticeable effect. My way of thinking about this is, if you have equal numbers of substrate molecules and inhibitor molecules, then there's an equal chance of either getting into the active site, and that would slow the enzyme reaction down by about a half, if that makes some sort of sense. So at the bottom of part C of question 4 then, says the breakdown by collagenase. Consider what kind of therapeutic drug could treat arthritis. And so they're looking for a drug that would stop collagenase activity, and essentially you're going to be saying an enzyme inhibitor. So you could use an enzyme inhibitor that fits into the active site of collagenase, and it could be a competitive inhibitor, fits into the active site of collagenase and therefore stops the breakdown of collagen, or it could be a non-competitive inhibitor. As we're going to discover, a non-competitive inhibitor then essentially does the same thing. It slows down enzyme activity. But this time what we say about the non-competitive inhibitor is that it doesn't compete with the substrate for the active site. We say that a non-competitive inhibitor may attach to some other part of the enzyme. So it doesn't attach to the active site, it may attach to some other part of the enzyme. And in so doing, it causes a change in the shape of the enzyme, and that causes a change in the active site. So you can't say specifically what non-competitive inhibitors do. You can say possibly three things. They attach to some other part of the enzyme that changes the shape of the active site and prevents the substrate fitting well into the active site. Or you could say that they cover the active site over and prevent the enzyme getting in. Sorry, prevent the substrate getting into the active. So sorry, just two things that either attach to some other part of the enzyme, changing the shape of the enzyme, or they cover up the active site. But I would always go in and answer. I always say that they attach to some other part of the enzyme, twisting the enzyme out of shape, that sort of idea, and making the active site like an incorrect shape for the substrate. Now, just looking then, so and there's elements of this in question 2 as well. So you'll see at the bottom of question 2, it asks. Question 2 is a really good question because it covers both the just fit theory idea and also inhibitors. It says enzyme inhibition ACE inhibitors. So back to the enzyme ACE. Describe and explain 2 ways in which these inhibitors may inhibit the ACE enzyme. And so you would say, well, these inhibitors may be competitive or non-competitive inhibitors, and then you describe competitive inhibitors as molecules that are similar in shape to the angiotensin 1 molecule that fit into the active site to compete with angiotensin 1 for the active site, and therefore they slow down the breakdown of angiotensin 1. That's a competitive inhibitor. A non-competitive inhibitor, a molecule that attaches to some other part of the enzyme, not the active site, maybe it attaches to some other part of the ACE enzyme, causing it to change shape, and therefore its active site to change shape and no longer fits the angiotensin 1. So that's enzyme inhibitors. Enzyme inhibitors are quite common in everyday medicine and biology. We come across them all over the place, and we don't recognize them as such, but things like drugs, medicines, poisons, chemicals that maybe damage the environment, toxins, etc. Lots of things act as inhibitors. A simple example might be take an antibiotic. An antibiotic kills bacteria. Well, it may kill the bacteria. It may not necessarily kill the bacteria straight up, but what it might do is an antibiotic may be an inhibitor for an enzyme of the bacteria, an enzyme that the bacteria depends on to reproduce or multiply. And by applying the antibiotic to the bacteria, then you damage their particular enzyme and they won't function. The same with lots of other poisons and chemicals that are used to kill organisms, toxins, and that. Often what they do is they act as enzyme inhibitors. Another example that you come across in other topics is the chemical cyanide that you're told stops respiration. You might have come across it with respect to it stopping active transport in cells. And it stops active transport in cells because it stops respiration. And remember, active transport required energy. Active transport requires respiration to happen, and there's an enzyme in respiration that is inhibited by the chemical cyanide. So cyanide is a very toxic poison both to humans and plants and animals, and because it acts as a non-competitive inhibitor, I think, for one of their enzymes that's needed in respiration. And you'll come across that possibly again at A2. So competitive and non-competitive inhibitors. On page 15, and looking at enzymes in medicine. Enzymes can act as biomarkers. This idea of a biomarker. Biomarker is really just a term that refers to some molecule or other that can be detected in the human body either say in your blood or your saliva or your urine, that can be detected, say, by doctors, that gives an indication of your health, or might indicate the presence of a disease or a condition in the body. Biomarker, so a marker molecule that gives some indication about the biology of the body. These biomarkers, the ones that you come across are mentioned in the specification there. There's one called elastase, and elastase is a chemical that's released whenever white blood cells are involved in dealing with, for example, lung infections. The white blood cells release a lot of elastase into the lungs, and that gets into the bloodstream. So the presence of elastase in your blood gives a quick indication to the doctor that there's something going on in your lungs, there's some sort of lung infection. Other conditions that throw this up would be your urinary infections there, urinary tract infection. There's an enzyme called esterase, and then there's one in heart attacks called phosphokinase. But there's lots and lots and lots of such enzymes that can be detected in blood and saliva and urine. And this is for that reason. That's why doctors often, when you visit to the doctor will often take a sample of blood or urine or saliva to send off to be analysed to look for the presence of these biomarkers. And often these biomarkers are enzymes. And then that allows the doctor to get some insider information about what's going on inside your body. Just looking on page 16 there, we have a bit of a discussion about enzyme inhibitors being used in medicine. So for example, aspirin is an enzyme inhibitor, a drug there to inhibit elastase. Penicillin is an enzyme inhibitor for an enzyme present in bacteria, and so on and so forth. That takes us then to a very important aspect of this topic that gets asked about fairly frequently, and that is immobilized enzymes. And so I've just written at the top of the section, I've written the relevant questions here, question 3, question 5C, and question 10B. And those are all, so that's question 3, 5C, and 10B. And those questions then relate to what are called immobilized enzymes. Now, immobilized enzymes is a rather unusual topic. It seems like something that you wouldn't associate with enzymes at all. But essentially, to understand immobilized enzymes, the best way to come at it is to understand that enzymes are very useful to scientists and very useful to scientists in many aspects of industry. And one of the most notable aspects of industry is in food production. Enzymes are involved in food production. So, and food production is a very big business, and anything that assists and makes the processes involved in food production faster or more effective then is a useful technology. So immobilized enzymes is essentially a technology based around enzymes. And the way I approach talking about, the way I sort of try to explain this, I'll give you a wee bit of illustration of what we mean by immobilized enzymes, is to think of it this way. This is really just a fairly simple illustration. In a normal enzyme reaction, in school, for example, if you're doing an enzyme reaction in school, you might be doing it in a beaker. Okay. And into the beaker, you might put the enzyme molecules. I'll just use the letter E for the enzyme molecule. So you've got the enzyme molecules, there they are. And I've used the letter E because you can kind of imagine that the active site of the enzyme is maybe in there or there, something like that. So that's the enzyme. And then what would you do? You would add into the beaker, you would add in the substrate molecules. So you'd put the substrate molecules in. So here's the substrate molecules, S, just using S for the substrate. And then you might stir the whole thing up, and you might heat it up. heat it a little bit, and the heat will give the enzyme and the substrate more kinetic energy, and they move around faster, more collisions, more enzyme substrate complexes, and so on and so forth. Then you would stir it. Why would you stir it? Some sort of stir. That's to ensure that the enzymes and the substrates are constantly in contact with each other. So the idea is to get them together to form enzyme substrate complexes. Now, that would be your normal process of using enzymes. You basically, and if you're doing this in industry, you simply would have bigger containers and a big container. And you'll see there on page 19, I've sort of illustrated that. I've said the traditional way of using enzymes was in a big what's called a batch reactor. So you just have like a reactor. It's just really a very big tank, and you put in whatever the molecule is, the food molecule, and you put in the enzyme, and you stir them up and heat them up and so on, and the reaction happens. Just for a moment, to give you a tangible example of where this may be used, you might know that babies get little tiny bottles of baby food. You know, little tiny bottles, if you know what I'm talking about. They're tiny little bottles of baby food, and they're for infants, for babies possibly under a year old. And what they are is they'll have all sorts of different flavors and different varieties. But essentially what they are is food that has already been pre-digested. So the food has been digested because enzymes were added to it to start breaking it down. It's simply to give the baby food that is already mostly digested, so that their little digestive system doesn't have to work too hard to get it digested. So these pre-digested baby foods. And lots of other foods that we purchase as adults have been treated in some way or other by enzymes that we're maybe not aware of, maybe to break the food down to some extent, and maybe tenderize it. Meats, there's examples of adding enzymes to meat products to sort of break the meat down a bit, make it more tender and juicy, as it were. Yeah. So anyway, in traditional techniques over hundreds of years, you simply had to add this, put the enzyme in a big container, and you put the substrate in the big container, and you let the reaction happen. Then at the end, you might open up a tap at the bottom or something, and you might let the stuff come out of the tap. What would come out the tap would be the enzyme. The enzyme would come out, and the product would come out, because the product would, and after a while the substrate would disappear, and you would end up with enzyme and product. Is that right? So you now would have enzyme and product. The substrate is gone. And essentially you open up the P for product, you open up the tap, and out comes the enzyme and the product. And this has been the traditional way of doing this. But one of the things then you had to do was, if you wanted to get a product, you want to get the product purified, you had to purify, you had to separate the enzyme from the product. And that was a process, this whole process is a bit time consuming. You have to stop, you have to empty out the mixture, you have to separate the enzyme from the product. And then if you wanted to do it again, you have to then put the enzyme back into the container and put in more substrate. So it's a very stop-start process. Industry doesn't like stop-start processes. It doesn't like processes that are time-consuming or expensive. And all of these things are expensive. So what industry prefers is some sort of process that can happen continuously. So what I've said there, 24-hour operation type process. Industry likes this kind of operations. So if you look at the bottom of page 19, what I've listed there is the disadvantages of this kind of process. I've said it's non-continuous. I've said after each batch the container has to be emptied and then refilled with more substrate and more enzymes. Purification has to take place before you get the product. You have to separate the enzyme from the product. That's time consuming and expensive. And also every time you do the process you lose some of the enzymes. You don't have all the enzyme molecules to go back in. They get damaged or they get lost or whatever. So it's all together a full process. Now, science then we're looking at ways of modifying this process. And what they came up with was something that does occur in nature, and you maybe would come across it later on when you discover that the proteins can enzymes inside a cell some of the enzymes inside a cell are not free in the cytoplasm. They're actually attached, or they're part of the cell membrane, so they're kind of stuck to the cell membrane. So let's just illustrate this. So this then falls under the heading of, this development in technology falls under the heading of enzyme immobilization. Enzyme immobilization, and as its name suggests, the enzyme is immobilized. This simply means it doesn't move around. It's stuck or attached to something. And what I'm going to illustrate very simply, this time I'm going to put the enzyme, I'm going to attach the enzyme to the wall of the beaker, and the wall of the container. So there's the enzymes, this time attached to the inside surface of the container. Now this is just for illustration's purpose. So there's the enzyme molecules now are attached. You can see them attached there. And once again, you can add in the substrate molecules. So here comes the substrate molecules added in. Now what happens this time is that the substrate molecules, the enzymes can't move. They're attached to the wall, for example, of the container. They are immobilized, can't move. The substrate has to go to the enzyme, and the substrate has to go into the active site, and then the substrate gets converted into the substrate gets made into the product. And the product then is going to open up the bottom here and leave the bottom open like that. So the product then can escape, or it can simply leave the container, simply goes right through, or I suppose it could come right through the top. But essentially what's happening here is that the product simply just can leave the container. So you just, essentially what's happening is you're pouring the substrate in at the top. Substrate's reacting with the enzymes, or meeting up the enzymes, and the product's coming out the bottom. And this can be a continuous process. You don't have to stop. You don't have to empty everything out. You don't have to separate the substrate from the product. 24-hour process. Simply pour substrate in the top, and product out the bottom. So this is enzyme mobilization. Now, the bit that I jumped a bit fast here is just telling you about the mobilization process. This is just an illustration. Mobilization is just some way in which the enzyme is physically or chemically attached to some surface inside the container, or on some sort of surface. Physically or chemically attached to some sort of, often the surface is described as an inert surface. Inert meaning that the surface has no impact on the reaction. And the surface can be plastic or glass, or even sand or fibers, paper, all sorts of things. And there are thousands of examples of this being used in industry now. So you can see how it's a big advantage. Well, you can imagine that it's a big advantage over the other process. And really, you're looking at the advantages, then essentially you take the original disadvantages of the other process and you just turn them around, and you can say, this process can be continuous. There's no need to stop. There's no purification. In other words, you don't have to separate the product from the enzyme. The product simply comes out the bottom of the container and it's released, and it's pure already. And this makes it a very fast, much faster process, and you don't have the expense of the purification. And then there was a third unexpected advantage of this. It was discovered when you attached, in many cases, when you attached the enzymes to the surface, or you immobilized the enzymes, it made the enzymes more stable, more thermostable, more pH stable. In other words, the enzymes weren't so likely to be denatured now in this immobilized situation. Because what explanation could we come up with that? Well, apparently one argument could be that when you attach the enzyme to a surface or some other material, the actual attachment of the enzyme to the surface, the fact that it's bonded to the surface, makes the enzyme structure stronger, stabilizes the enzyme, makes it less likely to be damaged if you increase the temperature. less likely to be damaged by unusual, if bonds do break in the enzyme is still sort of held in place and its shape is maintained. So that's the argument if you get asked about that. In what sense, in what sense is the enzyme more stable? You'd say the enzyme's shape is less likely to be damaged as the shape is supported by the support material, and this makes the enzyme more active over a wider range of temperatures and pHs. So this was a definite advantage. You could have high temperatures here. You could pour the substrate in at quite high temperatures, and that didn't denature the enzymes. The enzymes worked perfectly well, and higher temperatures then just made everything happen faster. So quite effective. And so those are the major advantages then. Now the question that relates to this, a recent essay question, I think the essay question is, I think it's maybe 10, question 10 if I've got it correct. Sorry. Yes, question 10, the second part of question 10, you were given 9 marks to describe enzyme immobilization. You're asked to describe methods of enzyme immobilization and give an account of the advantages and disadvantages of enzyme immobilization. So you're simply asked what are the advantages and disadvantages. Now we're going to come to the disadvantages, because it was also discovered there were certain downsides to this. And one of the major downside was, in some cases, the enzyme was damaged by the immobilization process, or in some sense, the enzyme didn't work as well as it would have if it was free in the solution, if it wasn't immobilized. And I've asked the question at top page 1. I said, why may immobilization reduce enzyme activity? And here's a good argument. Some active sites may be blocked or facing into the support materials so the substrate can't get at them. Let me just illustrate that. There's an enzyme there, and you can see the enzyme, its active site is facing out. What would happen if instead of attaching that way, the enzyme attached the wrong way around? So it attached to this, when it attached to the surface, it attached with its active site facing into the surface. And that would mean that the enzyme couldn't get access to the active site because it was stuck against the surface. And that was this is a good way of explaining why the enzyme activity might be reduced. A proportion of the enzymes, when they attach to the support material, they attach the wrong way around. Attach in such a way that the active site is blocked. And that means the substrate cannot enter the substrate cannot enter the active site. And in other cases, immobilization also might change the shape of the active site, so less substrate molecules can bind to the active site. So there's a couple of things. So the enzyme might face into the support material, or the mere attachment of the enzyme to the support material may damage its shape. So those are a couple of disadvantages. Now that question asks you to discuss different methods of enzyme mobilization, and there's no way I can draw anything on the board here. And just to say that at the bottom of page 21, there's a good table there that summarizes the major methods of enzyme mobilization that you need to know. And for each one, you have to be able to describe briefly what the process involves. So I'm just looking at them here. Adsorption, where the enzyme is attached by weak forces to an inert support material, and so on. And then you have encapsulation, entrapment, and cross-linkage. Now you'll notice in that table, I've got something here about advantages and disadvantages. Now those advantages and disadvantages are really comparing those different methods of enzyme mobilization, one with the other. So I'm sort of looking at each in turn and saying, well, this might be the best method for such and such a reason, and so on. So those aren't the general advantages and disadvantages of enzyme mobilization. Those are specific to those different individual methods. The other, just refer you also then to a very common question on this topic is, I guess, question 3. Question 3, and also question 3 and question 5C. Those are very common questions. Question 5C, in fact, they're so common that I put question 5C, put the graph for question 5C into the notes there because that graph comes up a lot. So what are they asking there? Basically they're saying Oh, it tells us there are enzymes in biological washing powder. So that's an example I forgot to mention. You know there's enzymes in washing powder. So the enzymes are adsorbed onto the surface of little particles in the washing powder, and that's the way in which they're immobilized. And this graph shows the activity of an enzyme over a range of pHs. Well, we've just said that one of the effects of mobilizing an enzyme may to make it more stable with respect to unusual pHs or extremes of pHs, and you can see that from the graph here. You can see generally there appears to be an optimum pH for this enzyme. It's around 6.5, and you can see both in the immobilized form, which is the dotted line graph, and the free enzyme, that's the non-immobilized. You can see that's the optimum pH. Now, it says it has described the main difference in enzyme activity which result from mobilization. So there's 2 marks there. So what's the main difference observed in that graph? Well, 2 things to notice. Notice that the activity of the immobilized enzyme is lower at the optimum pH. You go to pH 6.5, you see the immobilized enzyme has a lower activity. And if you were to explain that, you might say, oh, that may be because some of the enzymes, during enzyme mobilization, some of the enzymes are damaged, the shape is damaged, or some of them are facing into the support material so that the substrate can't get into the active site. The other thing to see from that graph then would be to say, oh, at extremes of pH, in other words, if pH is away from the optimum, and now it's away towards 9 and away towards 4.5, you can see that the immobilized enzyme has a greater activity. And there you would argue, you would say the explanation of that is that the enzyme immobilization has given the enzyme a more stable structure. And you'll see in page 22 there, halfway down the page on the right hand side, I've said that mobilized enzyme has a more stable structure. If you wanted to add there, you could say its shape is stabilized, and therefore it's less likely to be damaged by high temperature, by unusual pHs. So the same idea then applies to temperature as does pH. So there we are. The only part of that this topic that I'm not doing then is the practical bit at the end of it, the colorimeter, using the colorimeter. Now that turns out to be an extremely important aspect of enzyme work. I'll leave that for a separate video, possibly where I'm going through all of the practical aspects for module 1. So there you are. Hopefully that was helpful. As I say, if you need the question booklet, get in touch with me at that number up there, and I'll get the materials to you. And also, doing questions is extremely important. So it's useful to do as many questions as possible. If you get in touch with me, I'll get you a whole big bunch of questions and answer schemes and everything, and a set of notes. Okay, there you are. Thanks very much. Goodbye until the next time.